Good afternoon. I'm going to talk to you about how song mediates the extent of reproductive isolation between tinkerbirds at contact zones. So what happens when two closely related species come into contact? Well, we've been studying this in two closely related barbets in Africa that come together at several independent contact zones. Our study species are the yellow-fronted tinkerbird, Poconulus chrysoconus, and the red-fronted tinkerbird, Poconulus pazillus, and these two species meet in a number of contact zones in mostly the eastern part of Africa. What we're showing here are the distributions of the two species. The more widespread yellow-fronted tinkerbird in yellow, red-fronted tinkerbird with a disjunct distribution, shown in red, and the blue lines represent approximate locations of the contact zones where these two species come together, going from south to north in southern Africa, in Tanzania south of Lake Victoria, in Kenya and Uganda north of Lake Victoria, and then further north in Ethiopia. We've been studying patterns of variation in their genes and genomes, in plumage coloration, and also of most interest to this conference in song and its recognition, but I will talk about the other patterns of variation for background and context. So what happens when these two species come together? Well, let's have a look in southern Africa here. We have a red-fronted tinkerbird and a yellow-fronted tinkerbird. And in the contact zone, we might find some individuals that look like this, that have quite orangey foregrounds. Is that an indication that the two species hybridize? Well, we investigated this first across that contact zone in southern Africa, working in collaboration with Professor Aramondiam from the University of Eswatini and my former PhD student Emmanuel Nwankwo. A number of these sites depicted by the yellow and red dots showing locations where we sampled and recorded the two tinkerbird species across that contact zone. We'll start by showing here the substantial evidence that we have of introgressive hybridization between the two species using microsatellite markers. This is a structure plot. Each bar here represents the ancestry of each individual. The more yellow in the bar, the more yellow-fronted its ancestry. The more red-fronted its ancestry. The bottom banner here shows is essentially a transect between the distributions of the species. In green is the contact zone, and within that contact zone we can see that we have a lot of individuals with mixed ancestry, and there are mismatches between their genomic background and foreground colour, and also the mitochondrial haplotype, which is maternally inherited. If we look at the phylogeny, the mitochondrial DNA phylogeny, we can see that we have a number of individuals with a yellow-fronted haplotype, represented by this yellow section here, that have a red foreground, and that's all these red lines. And there's only one individual from this sample that was the other way around. So there's also evidence for greater similarity in foreground colour and symmetry than in allopatry in southern Africa. If we look at these panels here, we can see the box plots in the middle are the sympatric individuals of red and yellow-fronted tinkerbird. The outer box plots are the allopatric individuals, and so in symmetry, in brightness, in chroma, and in hue, those colours are more similar in symmetry than they are in allopatry. Now, usually when you have this much hybridization between two taxa, those taxa will have only very recently diverged. But in the case of these two, the subspecies that meet of these two species in southern Africa, they've been diverging for over 4 million years. So this is an unusually late case of such extensive hybridization in two species that diverged a very long time ago. In fact, according to this phylogeny, they're not even sister taxa. We've gone on to ask what the genetic basis might be for their colour differences. Might the foreground colour be explained by the same genes as carotenoid-based polymorphisms in other species? We used DD-RAD sequencing and assembled a genome of red-fronted tinkerbird Poconymus bacillus to perform a genome-wide association study. And we mapped our bacillus genome onto the zebrafinch reference genome. We focused on 3 plumage coloration patches, but I'm going to talk about the most prominent one, which is the difference in the foreground colour, after which the common names have been described. And this is based on samples across that contact zone in southern Africa. This is a Manhattan plot here showing which regions of the genome are significantly different between these particular traits that we focused on, ground score and hue in particular. And the genome-wide association study identified a cluster of SNPs on chromosome 8 that was associated with the gene CYP2J19. Have a little more closer look at this particular gene and the SNP that's closest linked, the SNP or variant that's closest linked to it. In the bottom panel here, we're showing the genotype on the y-axis at that locus against the hybrid index. So individuals with a hybrid index of 0 are red-fronted tinkerbird and 1 yellow-fronted tinkerbird. And what we can see is that at the locus, the homozygote for the red-fronted allele with a hybrid index of 0, these individuals have red foregrounds, and that's represented by the dot colour. Individuals that have yellow-fronted ancestry and have the yellow-fronted allele homozygous for the yellow-fronted allele, AA, they have yellow foregrounds or amber foregrounds. The heterozygotes that have a range of ancestry from red to yellow with a number of intermediates, those heterozygotes tend to be orange to red in foreground colour. And the top plot there is the hybrid index against interspecific heterozygosity, which shows what the foreground colour of, say, an F1 hybrid is, of quite reddish, and it shows the direction of the backcrossing, which is mostly back towards the yellow-fronted genome. And I'll talk a little bit more about asymmetric backcrossing later. We also looked at hue of foreground colour using spectral reflectance of feathers. And what's interesting here, so here we're illustrating the genotype of the CYP2J19 locus using the different shades. And the individuals that are, both individuals that are homozygous for the red-fronted allele and the heterozygotes have redder hues than all individuals that are homozygous for the yellow-fronted allele. And this is whatever the hybrid index, at the same hybrid index, you can see that they differ in hue based on the allele, based on the genotype at that locus. So that's pretty clear evidence of the role of this gene in foreground colour. Now, what do we know about CYP2J19? Well, it has been implicated as the catalase that catalyzes the conversion of yellow dietary carotenoids to red ketocarotenoids in birds with red feathers. And this has been based on lab crosses of canaries and looking at feather colour in canaries, and in zebra finches looking at bill colour. And it's also been extended to bill colour polymorphisms in long-tailed finch, Gordian finch. What we're showing in the tinkerbirds here for the first time is that it can explain differences in feather colour across a natural avian hybrid zone, and it's also the first time that it's been implicated in playing a role in yellow and red feather differences in a non-passerine bird. We also needed to confirm really the presence of ketocarotenoids in the feathers, justify the work, and we used Raman spectroscopy to confirm the presence of ketocarotenoids consistent with a role for catalyzing the conversion of dietary carotenoids in red-fronted tinkerbirds. And we can see that if you focus on the right panels here, individuals with red forecrowns are represented by the red peaks, and yellow forecrowns by the yellow peaks. And these values, these Raman shifts, are consistent with values for certain ketocarotenoids in the red feathers and yellow dietary carotenoids in the yellow feathers. And that's individuals in allopatry and sympatry, consistent patterns. Hybrids have something intermediate. So I mentioned before that backcrossing is asymmetric. This is something that we can see by looking at the admixed individuals here across our population. Number of admixed individuals also pictured. They have more XY autosomal ancestry than on their Z chromosome, suggesting fathers had more posilis ancestry than mothers. What it suggests is that yellow-fronted female times red-fronted male is much more common than the reverse cross. And so when we find a mixed pair, which we thought we might have found here with my collaborator Alan Brailsford from UC Riverside, we might expect that the red-fronted individual is the male and the yellow-fronted is female. I think what actually transpired in this instance is that they're both males. So it wasn't actually a pair, it was just two males chasing each other into a net. And in fact, this red-fronted individual is, to my mind, clearly a hybrid, and I'll talk a little bit more about that later. So we also analysed DNA samples from across two other contact zones to determine the extent of hybridization. So that's the contact zones in Tanzania and Kenya. And here we see a file structure plot based this time on nearly 30,000 SNPs from our genomic sequencing that reveals differences in gene flow across those contact zones. So on the right here in Southern Africa, we can see that pattern that we saw before of lots of mixed ancestry. So lots of evidence of introgression. Some evidence of mixed ancestry and introgression in Tanzania, little or no evidence from Kenya, suggesting that there they don't interbreed. So what's going on? And we can also look at that from the perspective of principal component analysis, where we can see that in Kenya, yellow-fronted individuals are very distinct from red-fronted individuals. No sign of introgression there. And this is the yellow-fronted individuals from Kenya group with yellow-fronted from the other side of Africa, from Kenya, from Ghana and Nigeria in green. Red-fronteds from Kenya and from Tanzania in orange, they are much closer to yellow-fronted from Tanzania. And there's evidence of introgression. We have individuals here with red foregrounds that cluster with the yellow-fronteds, an intermediate individual as well. And if we look at Southern Africa, we can see almost a genomic continuum here. We have all these triangles here. These are red foreground birds that are clustering yellow-fronted, extending beyond into the allopatric red-fronted population. So hybridization involves externi, despite its basal position in the mitochondrial DNA T. So this yellow-fronted individual here that's basal, this is the one that's hybridizing with both red-fronted populations in Southern Africa and East Africa. Whereas yellow-fronted in Kenya and Uganda is apparently not hybridizing with a phenis from Kenya and Uganda, and they've been diverging for a shorter period of time. What might explain this difference? Is it something to do with pre-mating? or is it post-zygotic isolating barriers? We're going to focus mostly on pre-mating isolating barriers from here and the potential role of song. So with Emmanuel and Ara and also with Kim Ortega, we recorded songs of these species across the contact zone in Southern Africa. So the songs of these tinkerbirds are remarkably simple in structure, both in temporal and spectral structure, and they're also very similar between the two species, as we can see from these spectrograms and waveforms, and you'll be able to hear from this recording of yellow-fronted tinkerbird here. And from this video of red-fronted tinkerbird, both these recordings from Southern Africa. So you probably heard there that the red-fronted tinkerbird sounded very slightly faster than yellow-fronted tinkerbird. We have focused on these two measures of the songs. One is the pulse repetition period or inter-onset interval, focusing on the period between when one pulse starts till the next pulse starts. So that's the inter-onset interval. And we've also looked at frequency, and we've measured this for essentially every pulse on all our recordings. Very briefly, in terms of frequency, there are no differences between the species. They overlap throughout as we go in each of these panels from the right in allopatry towards the left to the contact zone. And while there are no differences in peak frequency, songs do differ in pace, with patterns varying among contact zones. So we look at the right panel, we can see that in allopatry, as we go towards the 3 here, we can see they're quite different in pulse repetition period, but as we go towards the contact zone, we find a lot of overlap in song pace. In Tanzania, again, there's considerable overlap as we move towards the contact zone. In Uganda and Kenya, however, in allopatry, they are different in pulse repetition period, and they remain different as we go towards the contact zone. So there's no overlap in song pace. In Ethiopia, they're again quite different in allopatry and remain quite different as we go to the contact zone, but there are a few individuals here with red foregrounds that appear to be singing a song that's typical of yellow-fronted tinkerbird. So very little bit of overlap there. I'll point out that the axes are a bit different here on each of these panels. So if we take our gap analysis or the distribution of the inter-onset intervals to the level of distributions, we can see that the overall distributions of these inter-onset intervals differ quite a bit between the contact zones, so that the peak of the distributions between the two species is much more similar in Tanzania and in South Africa and Eswatini than it is in the other two contact zones. So we have substantial overlap in those that have these more similar distributions, and little or no overlap across the contact zones where the main distribution of the song and inter-onset interval is more different. And this might provide the context for species recognition if those differences are great enough so that they recognize each other as different species. Representing these inter-onset intervals geographically using inverse distance weighting, we can see that we have differences in clines across these contact zones. So in East Africa, across the contact zone in Kenya and Uganda, we can see a steep climb from the slower yellow-fronted tinkerbird songs to the faster red-fronted tinkerbird songs. Similar pattern in Ethiopia. In Tanzania, we see a very gentle gradient from red-fronted to yellow-fronted tinkerbird songs. And in Southern Africa as well, it's a gentle gradient between red-fronted to yellow-fronted tinkerbird songs, which are much closer to each other. In addition, so those analyses are based on the mean values per individual, but we're also looking at the distributions per individual using the 115,000 plus inter-onset intervals that we worked out and calculated, and we're calculating pairwise symmetrical Kullback-Leibler divergences between individuals across each contact zone. So these are the distributions of the inter-onset intervals of each individual, and we're looking at each comparison, and what we're planning to do with that is compare that divergence with the geographic distance, the minimum distance to a contact zone for each pairing, and see how that corresponds across each contact zone. This is work, and that will allow us to produce figures like this. And it's work that we're doing in collaboration with Florencia Noriega at Code in Berlin. So the question is, are the song differences recognizable across each contact zone? Well, we performed playback experiments to test this. What we found was that across the contact zone in Southern Africa, neither yellow-fronted tinkerbird nor red-fronted tinkerbird responded any differently to conspecific versus heterospecific playback. In Tanzania again, there were no differences in responses of yellow-fronted tinkerbird nor red-fronted tinkerbird to conspecific versus heterospecific playback. So it appears that they don't differentiate between the songs. However, in Kenya, both yellow-fronted tinkerbird and red-fronted tinkerbird responded significantly more strongly to conspecific playback than to heterospecific playback. Evidence that they differentiate between the songs, and we expect this is based on the pace of the song. So to conclude, differences in song similarity among contact zones may mediate reproductive isolation, thus providing insight into processes that generate diversity and maintain species limits. Now, plumage divergence may also play a role in maintaining species limits in Kenya. That's not something that I've really touched on in this talk. But preferences for red feather coloration may otherwise promote asymmetric introgressive hybridization. And so this is the situation where we have that asymmetric backcrossing in Southern Africa with those female yellow-fronteds mating with male red-fronteds. And this may indicate some differences in preferences for red feather coloration versus yellow feather coloration. Current and future work on this project. Okay, so speciation genomics. We're looking to identify, in addition to what we've done with feather color, is identify the genes mediating species differences with a GWAS on song variation. We're assembling a high-quality reference genome. So this is something we're doing in collaboration with Eric Jarvis at Rockefeller University and Bridget von Holt at Princeton. And also working on this is my PhD student, Sifiso Lukele, who's worked with me in the field in Eswatini and also in Ethiopia, and is involved in this study. We're investigating the ontogeny of song in these tinkerbirds and whether auditory feedback may play some kind of role. So tinkerbirds are non-passerines, they're pisiforms, and their song is thought to develop innately. However, this has not been confirmed experimentally, so it's something that we hope to do with captive breeding of red-fronted and yellow-fronted tinkerbirds, working with Aramonidium and also with Colleen Downs at the University of KwaZulu-Natal. We're also investigating song stability across the hybrid zones. Might hybrid songs be less stable, and might this help maintain species limits? Well, we're investigating this with Alex Hupflus and his PhD advisor Niels Dinkemanse at Ludwig Maximilian University, and this is something that my PhD student Matteo Sebastianelli is also working on. And this is the idea. So hybrids like this bird here, this orange-fronted individual singing this song, so is that song as stable as a parental form? Well, we can look at the distribution of the inter-onset interval of these are of a number of individuals that we've ringed and subsequently recorded singing. So that was one of them. And if we look here at the distribution of its inter-onset intervals, we can see it's quite variable. And there are some other individuals that are quite variable and others that are quite stable. Indeed, if we look down here, we can see another individual with an extraordinarily variable song. That is the song of the individual that I showed you earlier when I had the two birds caught in the same net, and I said that that reddish fronted individual was a likely hybrid. Well, that's that bird's song. We're also looking at this with, we plan to look at this with peak frequency as well, and variability in peak frequency. Some preliminary results. We can see that intermediate songs are more variable. So what we're looking at here is mean inter-onset interval per individual and their coefficients of variation. So more variable songs mean less stable songs. And what we can see here is that in Pacillus in blue, the slower Pacillus songs, that's these higher IOIs, are more variable, whereas in Chrysoconus we see the opposite pattern. It's the faster songs that are more variable. So intermediate songs between the two species are the most variable, suggesting that hybrids have more variable, less stable songs. We've been using double hierarchical GLMs to model the songs and the variation in these songs between the species. And using a quadratic regression function here, we can see that this best represents the pattern of variation between the two species here against what we're calling proportion Pacillus. This is actually based on a discriminant function analysis of the songs. Those most intermediate songs are the ones that are more variable. And this is across the Southern African contact zone again. We've also done it individuals that in those individuals that we ringed, focusing here on hue as a proxy for the hybrid index. And again, the data are best represented by a quadratic effect, showing that the intermediate value individuals those have the more variable songs. The plan here is to replace hue with the actual value of hybrid ancestry from those individuals that we've recorded, and we hope to be able to do that fairly soon, as we've just got the genomes in from those individuals. Okay, so the focus of my talk so far has been on interactions in the savanna tinkerbirds. I'm going to talk a little bit now about the forest tinkerbirds. So the savanna tinkerbirds have these ranges that abut each other. The forest tinkerbirds have largely overlapping ranges represented by these hatched lines here. And that's yellow-rumped tinkerbird, Pogonulus mediates, and yellow-throated tinkerbird, Pogonulus subsulphureus. The songs of these two species are more different than the two we heard earlier. This is a yellow-throated song. So again, we're talking about a pulse repeated at constant frequency, much faster than the other ones, and delivered in bouts. And yellow-rumped tinkerbird. So a slower song and typically lower in frequency. And we studied the songs of these two species in a number of populations in Simpatry and Alapatri in Cameroon and Uganda. This is work that I did with Dan Blumstein and Tom Smith from UCLA. And what we found was that the Sympatric populations sung songs that were more different in frequency than the songs in Alapatri. And the argument for this is that based on our playback experiments as well, is that these greater differences in frequency help them recognize that they're different species and they respond less to each other's song in sympatry than they do in allopatry. And so the idea is that this helps reduce interspecific aggression in sympatry. We've since gone on to ask the question of how long they've been interacting and how the patterns vary between the populations that we studied in Uganda and Cameroon and then in West Africa, where they meet again. We found a clinal pattern variation in song in both species. We're going here from East African red to Central African green, West Africa in blue. And what we see is that as we go from east to west, the rate becomes a lot slower in yellow-throated and to a less degree in yellow-rumped tinkerbird, and the frequency becomes higher as we go from east to west in both species. So that in West Africa, the songs of the two species are much more similar in rate, but a little more different in frequency. Having said that, looking at the songs of two species in sympatry and allopatry, or what we're calling here, where the congener is present in low or high densities, we can see that the clear pattern of character displacement in the song in Central Africa, compared to where they occurred together or in low densities or in allopatry, and to some extent that pattern in East Africa, but the pattern does not occur in West Africa. What's going on in West Africa? Well, I can start by saying that they don't overlap. The ranges don't overlap. They're largely allopatric. They abut in a similar way to the savanna tinkerbirds, whereas they overlap so much as we showed before in Central and East Africa. So what explains that difference in their distributions as well? Well, we can look at the mitochondrial DNA phylogeny to find what explains it. And the phylogeny supports longer interaction times in the east compared to the west. And what we're doing here is we're looking at the how long the different populations or subspecies of each species have been diverging from each other on either side of the Dahomey Gap. So the Dahomey Gap is an area of savanna that separates the upper Guinea forests of West Africa with the lower Guinea forests of East Africa. And we can see that the yellow-throated tinkerbird here has been diverging either side of the Dahomey Gap for over 1 million years. It's been present on both sides for at least 1 million years. Yellow-rumped tinkerbird, on the other hand, has only been diverging either side of the Dahomey Gap between 200 and 300,000 years. So it's been present on both sides only for that long. So the question is, which side has the yellow-rumped tinkerbird been on longer and interacting with yellow-throated longer? Well, the evidence from here, based on some of the deep splits we have in East Africa, shown in red here, is that it's been present in East Africa a lot longer, and it's only recently diverged and expanded its range, into West Africa. So what that suggests is that it's been interacting far longer in East Africa with yellow-throated tinkerbird. That length of time that it's been interacting has given it the opportunity to diverge in certain characters, such as song, which facilitates their coexistence. In West Africa, they've been interacting far less time and not long enough to be able to diverge in characters that will facilitate their coexistence. But the age of divergence does not always correspond with phenotypic differences. This is something that we see between different subspecies of yellow-rumped tinkerbird. So, as I showed before, or as I played before, this is the song of yellow-rumped tinkerbird from Cameroon. And here's a yellow-rumped tinkerbird from South Africa, different subspecies. So very similar. Look at this spectrogram of the song of yellow-rumped tinkerbird with the fisher-eye subspecies from coastal Kenya. You can see straight away that this has got a very different temporal structure. In fact, it looks faster than the yellow-throated tinkerbird from Cameroon. And I will play you that song in this video. So obviously it's a very different song. Yet, fisher-eye, which occurs in Kenya, as we said, coastal Kenya, and also in Zanzibar, has been diverging from those Southern African populations and Tanzanian populations far less time, that's 500,000 years, than other populations of yellow-rumped tinkerbirds have been diverging. And in spite of that short period of divergence, those songs are different enough so that we get clear differences in responses to playback experiments, where responses are far stronger to conspecific song than to heterospecific song, both in yellow-rumped tinkerbird of the nominate Balaenata subspecies and of the fisher-eye subspecies. Significant differences in their responses, suggesting that if there is a contact zone, which there might be, but we've not found it yet, then these two subspecies, as presently recognized, would be reproductively isolated from each other. And so you can contrast that, this short period of divergence, rapid song difference having evolved, with a situation in red-fronted and yellow-fronted tinkerbird, which have been diverging for an order of magnitude longer time, yet they hybridize extensively because their songs are so similar. We've been working on a number of other studies in bioacoustics, both with tinkerbirds and other species. We've explored the effects of noise from the ocean on tinkerbird song, and that's something that Matteo Sebastianelli presented on earlier today with his talk. Work in collaboration again with Dan Blumstein from UCLA. That's Matteo. And we're also going to hear a talk on Thursday at 10:35 from Besong Bikeng, who's been studying the soundscapes and acoustic competition in the Amarum Forest Reserve at Plori, the AP Levenson Ornithological Research Institute in Nigeria, where she's been looking at differences between sites where she's recording using song meters, with differences in habitat, so in the gallery forest, the rocky outcrops, and the savanna scrub. We've also been working on non-vocal song, a study that we did with Chris Clark and Rick Prum from Yale, and Luis Juan was a former student from my lab. And we were investigating the mechanism that produces this sound during this display. So that's that loud wing song is produced by aeroelastic flutter of primary wing feathers. We investigated that using high-speed video, as we can see here, and synchronizing that with audio recordings. And we also performed experiments in wind tunnels and also some manipulative experiments with feathers to see how the sound was distorted. So that's non-vocal song in broadbills. And with that, I'd like to acknowledge many collaborators who worked on this project. A number of them are pictured here. Some I've not mentioned that worked with doing a lot of the genomic lab work, like Michaela Moisi, Bridget Okolowa, Sion Asfor, collaborator from Ethiopia, Greg Grether and Nadia Seal, who've worked with us on plumage coloration in feathers and the differences between the species, and Alan Brailsford and his student Daniel Pierce, who worked with us on the genomic analyses. And with that, I'd like to thank you as well for watching and ask if you have any questions. Hi.
Hello, hi, how are you?
Good, thank you.
Great, thank you so much. And actually, it's so nice to see all the different studies that are going on and that you've been working on. I'm just going to quickly say that tomorrow morning we actually also have a talk by Froen Beaker, which actually does a review of the different bioacoustic work in Africa. And I think that a lot of your work will be featured there as little counts in the bird work. And yeah, so just a little update there on what's going to happen tomorrow with that review. I have some questions for you that have come through, so I will fire them off. I'm not sure if this is completely biased, but I asked a question and it got 4 votes, so I'm going to read it out. And my question is it common to have species where differences are apparent in the pace of the song, but no differences in the frequency?
Is that common in birds, or is it common in
birds? Yeah.
In a lot of the... What I can tell you is that certainly in tinkerbirds and some of the other birds that we study, we find often that there's a significant correlation between body size and song frequency. Usually a negative correlation. And that seems to be the case in tinkerbirds as well, particularly as birds that develop their songs innately. And because of the extent of overlap in body size, we find that the frequencies don't differ in the songs of the 2 species that I started talking about at first, red-fronted and yellow-fronted tinkerbird. In yellow rumped and yellow-throated tinkerbird, I didn't include some morphology slides there, but we do have body size differences, and they relate to the song frequency differences in those two species.
Okay.
Yeah.
So my next question is, it looks a bit like you had a bimodal distribution of the intercall duration in the Ethiopian population of red-fronted. Are those individuals that overlap with yellow-headed, possibly hybrids?
Very possibly. We've just been getting some of our genomic data back now from Ethiopia. This is fieldwork that was done last year. So we hope to confirm that soon. My expectation is that hybridization does occur across that contact zone, but that it's much more limited than it is in the other two. That would explain the patterns that we see in the songs as well. But there could be very limited hybridization in Ethiopia.
Okay.
Yeah.
Okay, moving on. It's a comment from Gilly here, and he says, "Very nice talk, congratulations. What about the hybrid zone? Sorry, what has the hybrid zone to tell us about the species conservation and habitat loss?"
So it's important to really understand what the habitat preferences are of the species, and to see whether habitat loss may play a role in bringing some of these species together. This is something that we thought may occur in the forest tinkerbirds, that the habitat loss there was leading to more interaction in forest edges, secondary forest, that sort of thing, between two species that have otherwise different habitat preferences. But they don't hybridize. In the two species that do hybridize, it's not evident to us that they're coexisting, or they're coming into contact in an area that's degraded. I mean, yes, you know, the contact zone that we work in it's almost the reverse pattern. The contact zone that we work in Eswatini, the habitat is pretty good there. It's ranch land. There's grazing, that sort of thing, but there's extensive habitat that the species coexist in. In Kenya, the contact zone, I mean, the habitat there's just been destroyed. It's all agriculture, so we don't find them together much at all, and they don't hybridize. So I can't see an explanation that it could have some effect on conservation from that perspective. Mm. But it's something to potentially explore in future.
Okay. I'll just wait for that. Okay, so I have another question. So it says, it's from Andrea, and she says, "Apologies if I missed this, but are all of these calls related to reproduction? Have you looked at the potential role of these calls in sexual selection? For example, if females or males prefer slower or faster calls, this might affect the ability of species to interbreed, and the preferences could vary in the different regions." There's 3 questions there.
Yes, it's not something that we've really looked at so far, but it follows on from some of the work I think that we're doing now. We're looking at song stability. It could be that individuals with more stable songs are preferred than those with unstable songs, and so that may help mediate reproductive isolation in the contact zone. But in terms of sexual selection, actual preferences, that's not something we looked at yet, but with our plans for experiments in captivity, we are hoping to look at sexual selection, mostly in plumage coloration, but there's potential to also do experiments with songs as well, and see how females respond to those. I don't know if I answered all those questions. There was also about the comparison across the contact zone. So one of the things I didn't mention in the talk was that in Kenya, where they appear not to interbreed, the plumage difference between two species is greater there, in that the yellow-fronted tinkerbirds have brighter yellow underparts, and it could be that also plays a role in mediating reproductive isolation across that specific contact zone. Okay. Assortative mating.
Yeah. Wow. Okay. This is a follow-on, I think, from the earlier question. Sue-Ann asks, when you say the frequency correlates with body size, is this also true of song rate?
I don't think that we've seen any relationship so far. I don't know that we've really tested for it yet, but any relationship between body size or any biometric measure with song rate. It's something that we can explore for sure. We have a lot of data across a number of different species in the genus that we can investigate that with. For those that saw Matteo's talk earlier on the effect of surf sound on the frequencies, we've not looked at that yet with rate, but again, that's the pace. In a noisy environment, you might expect a faster rate, and that's something that may explain in that study the trill song that you find in the coastal populations, as opposed to the pulses of birds more inland. But yeah, we should also look at the relationship with biometrics. The tricky thing is, and this is something we're working on now, the tricky thing is with these tinkerbirds is relating your biometric data and your genomic data as well with the song itself. You need to be able to record the birds that you've color ranked, and this is something that we've started to do when we've done this for about 30-40 individuals, so we might be able to identify some patterns from that, but that's a lot trickier than say looking at plumage color, where every bird that you catch you can take some feathers.
Yeah, I see a lot of overlaps here between the studies that we do on dolphins. I've definitely, in my own experience, can see that the smaller, the smaller species of bottlenose dolphins makes calls and loops faster and things like that than the slightly larger bottlenose dolphin. But interestingly, in that scenario, the larger bottlenose dolphin makes higher frequency sounds, which is also kind of strange. But that's
well, it relates to some of the things that David Ruby talked about, I guess, earlier today.
Okie dokes. I'm just going to check on whether we have any other questions that come in. Okay, let me just check. No. Okay, so I think that's you off the hook, Alex. There we've got. Okay. I'm sure if anybody else wants to ask further questions, then there's the dedicated question and answer selection for that session, so you can ask the questions there. And then, yeah, absolutely. That just leaves me to say thank you so much for your talk.